take a look at this and say, oh, well, there's some things that you're gonna face that are gonna be tough in this chapter. And the reason they're gonna be tough is because now really we're really focusing on specific molecular events to describe how a muscle works. All right? And so these are things that you aren't gonna see, right, with your own eyes. The best you're gonna visualize is a model like this. The good news is there's tons of animations out there on YouTube. And you see this process in motion, or in dynamically, as opposed to statically, right? Like on my PowerPoints are static. And I can only go so far with gesticulating with my hands to get you to understand that, and I can only motion so much at the models and to try to get you to relate to it. So when you do watch these videos, and you really do want to, right? You're gonna have to pay specific attention to what's going on. You're probably gonna have to stop the video a couple times and like, what just happened there? And why did that happen? Because if you can lay the foundation here for this, right, cause and effect, this interrelationship between the nervous system and muscle here specifically, it's gonna be so much easier for you to grasp other things that we start talking about as you go on into AMP2 and you go on talking about the body. Because everything about the body is defining some relationship between two things or three things or more, and it's always cause and effect. So this thing happens and it leads to this thing and leads to this thing and this thing. So you're going to face a lot of sequential events, right? So a bunch of things happening and it cascades sometimes, right? And you're going to have to keep track of this new vocabulary, all right? So this is really the first chapter that we're hitting you with a little bit of physiology. Now, the really good news here, and this is not going to be good news when you go on AMP2, because it won't be true in AMP2, right, is that once you understand how muscle cells work down to the sarcomere, and that's all we need you to really grasp is the sarcomere, because you've got billions of sarcomeres in a muscle, right, varying in their size or varying in the number of sarcomeres, but they're all doing the basic same thing. So if I can understand what that one sarcomere is doing to lead to this event we call contraction, right, then I got it made. I'm just going to transfer that knowledge to every other muscle I'm studying and naming. And that's what I said. So if you look at these muscles in lab, and you're looking at the direction of, these are the fascicles you're seeing. So that was one of the more missed questions on the quiz, right, as well. It's just definition. So these are actually fascicles you see. And you would see those if you bought meat, right? My meat is muscle. And what you're going to see is then, if I could zoom into that fascicle, it would be organized, right, into a whole bunch of muscle cells we call muscle fibers. Muscle cells they're different because this is one of the big four tissues, remember that? And this tissue moves. This is the tissue that does all movement, all right? So it's going to have to be structured to do movement. So it turns out in skeletal muscle, what you have is you have a typical width of a cell at, on the micron level, maybe 20 to 50 microns in width, but long, up to 30 centimeters, all right, long. But again, you can't see them because they're so small in width. Extremely long cylindrical cells. You learn later on in chapter 10 that these resulted because in development, all these myoblast cells, because remember that blast is always a builder, always a former, had fused together. So that's why these cells they're multinucleated. And they're the only muscle cell of the three types, right? Cardiac, smooth, and skeletal, which you learned back when we talked about tissues in chapter 4. All right? They're the only ones that will have multinucleate, right? So lots of, because of this. And this just helps them with their function, right? So I want to really understand them down to this level. Most of your questions on your practical, when that comes up, is going to be, okay, here's a piece of tape on this, name this. Tell me what it does. Tell me what its origin. Tell me what its insertion is. I mean, again, it'll be one of those three. Right? You're not going to have, or one of those four. You're not going to get all four in one question, right? But you got to know all four because you don't know how we're going to ask the question. But I'll guarantee you it'll be identify the muscle, tell me the action, tell me the origin, or tell me the insertion. Okay? So that is straight anatomy. Right? But that action, again, we can begin to grasp, even if we can't, even if you don't have it memorized, like we said, we have a lot of information to memorize, because again, I'm looking at, ultimately, the way the cells are aligned when I look at the fascicles. And I know they're going to be shortening. So all I'm going to do is picture that whole muscle, right? Now, if I know the origin insertion, and you don't necessarily, you can take a pretty educated guess if you didn't, right? If I know the origin insertion, then I know the action. Looking at the fact that they can only shorten and pull, right? And that, right, I'm going to bring these 2 points, and sometimes more than 2, together or closer together, I can take a pretty good guess at what that muscle is going to be able to do, even if I don't know its name, right? Okay, so you want to start to kind of think about this. So here, as I promised, let's take a look at this pre-chapter just real quick. Don't write down it. This is not what I'm showing this to you for, all right? You know, this is basically, again, not surprised, 35, 15 to 65%, right? Very much in the low range. As we look at these scores, right? I'm not surprised. I'm not shocked at all, right? So we start off right away. I said fascicle. So there's definition. Which of the following defines a bundle of muscle fibers or cell? That is the fascicle, right? These parts are connective tissue that end in the mysiums, and the myofibril. Again, don't write down or take pictures of questions, right? But the myofibril, what is that? Well, that's this thing right here. Right? So this is part of a cell, and that is part of a cell. Right? So these are highly zoomed in aspects of these muscles you're talking about in lab. So the myofibrils themselves are the components, the units within here. But we're still not down to the unit, which is the sarcomere. Right? So, yeah, not surprised. I mean, again, because it's keeping these new terms. And by the way, you'll have fascicles in the nerves as well. It's one of those very common terms to anatomy. All right? This one wasn't so bad. You actually kind of getting the A bands, I bands, Z, H zones. These are all organization of the sarcomere. And we have to talk about this organization so we understand its function. So yeah, we'll use those models and say, and we'll put a piece of tape on there and say, okay, tell me what this is. And it was definitely the most missed question on my online quiz. I mean, I gave them, right, this model, and I gave them that model as pictures. And I said, hey, on my quiz, I said, what is this representing right here? And most of them told me the A band. I mean, that was the overwhelming answer. And that is, in fact, the sarcomere. Okay? Because I'm going from a Z line to a Z line, and that's a sarcomere. And as we talked about before class, the Z lines are the walls, more or less, of a sarcomere. Right? And yes, in fact, they are kind of zigzagged on here. And even if you look at it very closely, it says Z up there. Right? So, by the way. But they didn't have that advantage, right? Because they were just looking at a straight two-dimensional photo. Okay? And if I look at that model, and I'll come over to Jaden's, right? If I look at this model, of course, different model maker, different things I'm trying to emphasize. This I've kind of zoomed out. That's only got 1.5 myofibrils on it. This one's got like 7 or 8 on here, right? Cut at different levels to show different things. So the sarcomere is actually, on this case, is this big, heavy pink line to another pink line. Now it's not pink in reality, right? But that's the way they represent it on here. Okay? But these are all, again, myofibrils. And the myofibrils are made up of myofilaments. So we have all this organization and terms we have to keep track of in this chapter and in lab. Okay? So, right? So good. And again, the A band, these are German terms, and that's what the initial is actually, so that's why we just use an initial. And as I was telling you guys, the only one that really you might relate to is the M line, because M stands for Mittel, right? Mittel, M-I-T-T-E-L, which is middle in German, and it is the middle of the sarcomere, right? So the rest of them have to do with the way light is reflected, right, from the actual, as you take images of these. So they won't make sense to you. So using A, I, M, H is the way we've always represented. But what they are they're showing you the organization, all right? So what is the A band that you guys did get? Well, the A band is this area or zone, if you will, in which you have overlap between the thick and thin myofilaments. Actin and myosin are the main ones. So the A band is there. It's a good metric because the A band will never disappear in the contraction. The I band will. All right? And so these are ways that you could tell how fully contracted a muscle is when you take pictures with an electron microscope of muscle. So what's the I band? Well, the I band is where there's only actin. And so when a muscle is shortening, or sarcomeres are shortening, because we have to take that as sarcomere, and sarco means flesh, and mere is a measure. This is the measure of flesh, right? So what's happening here is that these Z lines get closer together. And so the limit of the contraction is actually at the A band, and the I band will go away, because you will lose the actin-only zones. Because what it's doing is being pulled, right, by the myosin in the A band towards the middle of the sarcomere. That's what a contraction is. And you just have to think about it for just a little while to get this. But this is why we value these terms. So an H zone is an area where there's, right in the middle of that sarcomere, really, where there's just myosin. And then the M, of course, is the very, very middle. So that's, we want to know and describe some of these aspects of the muscle. Not surprised that troponin is the calcium. So this is what we're going to strive for. I got to know what all four of those should do. That's what that question's telling you. All right? And troponin is going to be the calcium binding protein. And you got to give everybody a job. Okay? And then in this one, again, which of the following is an invagination or extension? Right? So it's, again, using imagination, picturing this. Well, this is the T tubules. And T or transverse, because transverse, they're going through. So the curiosity should be, well, why did you need, in a muscle, to do that? Why did the membrane out here, because that's what that is, have to come into the cell? I didn't need to do that in the epithelial cells, for gosh sakes. I didn't need to do that in the connective. Why is that important? And you'll notice if you look at that model, like what's on Jaden's desk and there by Tanner, look at all those little dimples in it. Do you guys see that? That wasn't the artist's rendition. Those are all little places where the T tubule would be invaginated, coming into, right? So that's going to lead all the way in. Now on that model, they show the T tubules as a big orange tube, right? On this one, they don't color code them. It's just this line right here. That's a T tubule. There's a little divot that it's coming in through. And they only show you a few of these on this model because, again, this is a more zoomed-in model. So T tubules are areas where, again, the sarcolemma, right, the membrane, because we give a lot of specialized terms in muscle cells because they're one of the big four tissues. They deserve some special terms. So the sarcolemma is our term for this membrane, and this, right, T tubule is an area where the membrane comes in, right, moves in to the sarcoplasm, which is the term we use for the cytoplasm. Okay? Yeah, Kale?
So, when I was talking about like muscles, in the first question, talking about what molecules and ions are needed. Are we talking like NaCl, epinephrine, like these type of things, or is it like going to be?
I'm sorry. Which are you talking about? Like
the first
quiz?
question of the muscle tissue group worksheet. We're talking about like what molecules or what like we had calcium, potassium. Like
yeah, so that's exactly what I'm talking about. So molecules in general would be something large. Ions would be the calcium, the sodiums, the potassiums. Mm-hmm.
Gotcha. Okay.
Yep. So like again, give everybody a job because you're going to need them. All right. So what of the following is considered the motor unit? Again, it was 40%. So a strict definition of a motor unit we're going to get to in class, and I'm just going to pass that up. And I'm going to say, look, that's going to be, right, you can't see it on these models because there's just one cell. But a motor unit, if you take that model, right, and I'm going to use this one up here, right, because it shows a little better on this. So this, again, this is a muscle cell. One cell, also known as a muscle fiber, right? And so this right here, that is, right, the axon of some motor neuron, which could be all the way back in the spinal cord, right? So this could be a muscle easily out here, down here, and the axons for this in the spinal cord, the cell, the neuron itself and its cell body in the spinal cord, and these axons will be very long, coming out, wired to the muscle cells. So when we talk about a motor unit, you know a unit is something that does everything together. So a motor unit is going to be the specific motor neuron. Again, you only see the axon endpoint here, right, which we call the end plate. We didn't need to show the whole thing. But that, right, neuron, motor neuron, because motor neurons do movement, right? That's why they get called motor, right? And all the cells that they would make contact with. So at the end of the axon, because the axon is just going to be one process from the cell, one and only one, okay? But when it gets to the close to where it's going to stop, it branches and it touches. It doesn't directly touch like I have David here. It comes extremely close because everything's separate in the body to muscle cells or fibers. And the number of muscle cells or fibers it contacts defines the unit. So motor units vary in size, don't they? So the fewest, just to give you a limitation, the fewest muscle cells or fibers that a given motor neuron can contact is 2. The most is 1,000. So, why? Why would that be the case? Why would we have this huge range? Because the fewer eye contact, the finer the movements I do. So here, my motor units in my hands are going to be very small. Because of what happens when a motor unit gets excited. So when that neuron tells, right, communicates with this axon, and we need to understand that muscle has to be told what to do. It does not do it on its own. It must be told to do the movement, and it's being told by the nervous system. That's why I said this is the first system we really have to look at this cooperation, and you'll have to see it from here on out in the body. Right? So when it sends electrical changes to tell that muscle cell to contract, it will tell every muscle cell it contacts, and they will respond. If everything is normal, they will respond. So in my larger muscles, I'm going to have bigger motor units. Because when I want to walk, I'm going to need a lot of response to move my bulk through space. But when I'm holding a pencil or using a tablet, right? Sorry. So I came a little too close, right? All right. When you're doing that, if you don't have fine motor movements, if you don't have just a few, you're going to be moving all over the place, right? You're going to be applying too much force for the situation. Because that's what's going to happen when these muscles start to contract. They're going to generate force. Okay? And that's what we'll always look at when we try to decide about how muscles are working. So it's extremely important that we get that idea. And here again, in this question, which will initiate or come first? Acetylcholine. And that's the answer to, one of the questions, the one Kayla's asked. That's definitely one of the very important molecules. And here's the good news. It's only going to be acetylcholine here. Because we have 40-some different neurotransmitters. And that's the term we use for these chemicals, right, that communicate between a nerve and a muscle. They're called a neurotransmitter, right? And they also let neurons talk to neurons. But it's chemical and electrical communication in the body. And here, when a motor neuron, as we talked about here, is telling a muscle cell to contract, and please understand, you are, right, in charge of the skeletal muscle. That's why we call it voluntary. So when you initiate this transmission of electricity, right, and we get down to the point where the neuron is contacting the muscle cell, part of the motor unit, well, acetylcholine is the messenger. It's the communicator. And so where is it on this? Well, it's these little yellow, they're little vesicles. It exocytoses from here, this motor neuron. So the motor neuron made this chemical, and they store it. And when the right situation arises, they will release it. So we're always going to start, at least in this chapter, with this idea of acetylcholine. Now, we can back it up and figure out what happened here to get to the point of acetylcholine, but we have to figure out some place to start. So let's start with the motor neuron tells a muscle cell, hey, I want you to do something, and that do something is going to be contract, right? Pull, because that's all skeletal muscle is going to be for us. And here's the way I'm going to tell you. So I always say to students, consider that pretty much number one. But these are just words on a page. That's why I said we have time together in class for me to show that to you, and you have time in lab to think about that. Except for Tanner, who told me already he's not going to learn anything today. So I'm kidding you, Tanner, right? All right, so I'm going to challenge you not to learn anything, Tanner. All right, so what happens when the acetylcholine binds? Well, this is going to be really important. Again, these are just words on a page. I see why you guys didn't do stellar on this, because it's hard to relate to. Even when you see it, it's going to be hard to relate to because this, again, it's microscopic, right? So sodium channels will open and sodium will enter. What? So here, right, in this area, in this sarcolemma, right, this membrane we have here, right, there are a variety of different proteins. We tried to bring that to your attention in chapter 3 when he said these cells in your body are pretty complicated. And we started talking about transport mechanisms, and we did that in lab as well. Well, that's going to haunt you throughout, right? Because it all comes down to really those principles, no matter what we talk about in physiology. Yeah, Kale?
Are there certain muscles that respond to, say, potassium more so than like calcium or salt, or, you know what I'm saying? Like there's
Not skeletal.
Not skeletal.
Skeletal is pretty simple, and that's why we start off A&P1 and skeletal, and we cover cardiac and smooth in A&P2. Okay, we want to give you the rules, and then we'll start to give you the exceptions. Okay, so here, those sodium channels, well, we would draw them in. They're not shown here. They're often not shown on cartoons. So some, the animations might show them. They might not. Some of mine do, and some don't. But right in this area, again, picture this white. Right here is the sarcolemma. And this darker orange is space between the end of the axon. And so this dark orange, again, is what we're going to call the synapse, where they come together, syn joining. All right? And then the white. So in this white, right, appearance here, you're going to see a bunch of sodium channels. But we'll back up one more. After the acetylcholine binds, it's going to quickly move across that space, very small space called the synapse, and it's going to attach to receptors. So again, remember that your membranes have all kinds of features to them. So in this muscle cell, we have to have receptors for acetylcholine, or nothing is going to happen. All right, how do we know this? Well, there's an autoimmune disease called myasthenia gravis in which the person's own defense cells attack and kill their acetylcholine receptors. And what do they suffer from? Paralysis. Because if you can't tell the muscle what to do, it's not doing anything. So we have to understand everybody's role. As I said, there's a lot of players. And we got to take it step by step. So acetylcholine is the communication between this neuron and this muscle cell. Acetylcholine has to be recognized by the muscle cell. It has to have receptors for it. It has to physically contact these. And when it does, that binding event, right, opens the sodium channels. Now, this wouldn't be important unless I understood that sodium is very high outside of a cell and very low inside. So you all remember from chapter 3 that when you have a concentration difference, it's always from high to low. So when sodium channels open, that sodium just go right in. It's like the door was closed. All of a sudden, there's a bunch of you out there, and I open it, and you all come flooding in. I mean, it's the same principle. All right? So that's our next event, right? Acetylcholine released, binds its receptors, sodium channels open, sodium enters. So that would be one of the ions, right, that we talk about. Okay? So we go a little further here. When does a contraction begin? Well, we're not there yet, are we? Right? I mean, honestly, we just got the rest, and this will happen very quick. It's milliseconds for all this to transfer, and that's why it's also hard for students or anyone to get their mind around it, because it happens so fast. All right? So I've got to get to a point where myosin can contact actin. It's not right now. It's not floating in this cell either, but the thick and thin lines, they're not directly contacting. So only at the point that I can get those two together am I going to get contraction, or I'm not doing any movement, I'm not generating any force, and I'm going to be a gelatinous blob on the floor. Y'all understand? Because you always have tonal contraction, even when you're not actually moving. Right? So I want to think about that. So how did I get to that point from the acetylcholine point? Okay? But the contraction is defined by myosin binding actin. That's the event that we're after. Or I'm not getting contraction. All right, here, which of the following would happen during relaxation? Because we all know if you contract, you really want to go back at some point. And even if you don't want to, you're going to be forced to, aren't you? You ever had fatigue? I was about a mile or so. I carried, in the States, it was an 8-pack of soda. I carried from a Hy-Vee up in Ohio because I missed my bus down to my apartment, right, down the road. And I was like, that was probably the most painful thing ever, because I'd been in a contractile state with that force, right? I had to overcome that force. And I was a poor college student. I didn't want to drop my glass bottles, right? You know, so that was probably the closest I came to just tears of I cannot move on, right? You know, and I mean, it seems ridiculous, but that's the idea. If you've ever carried groceries, if you're a parent and you carry your child, you understand what it's like to stay contracted. But even if you don't want to do it, you will have to let go of the contraction because skeletal muscle fatigues. Now let's take an opportunity to ask you about this. Does heart muscle fatigue?
Yeah.
It'll be the last time it does. Right? Does heart muscle fatigue? No. No, it does not. And we're going to have to look for why it doesn't. I mean, if I tell you it doesn't, don't just take my word. Why doesn't it? Does smooth muscle fatigue? Probably not. Because it's not, we don't put much demand on it, right? So I'm correlating our need, right? What do I want from this muscle with this idea of fatigue? So fatigue is going to be a point where if we classify it physiologically, I run out of ATP. Which, yes, believe it or not, it can happen. It's hard to do, but it can happen, right? Usually we feel the signs of fatigue, which is weakening contraction, and we stop the contraction. Okay, because I'm going to convince you, hopefully in this class, that if you run out of ATP, you are not contracting that muscle, right? And you are not relaxing this muscle. So this will result in what we call physiological contraction, and you're just going to fall to the ground, right? Because you're not going to be able to move that muscle, because you're going to need ATP to get contraction and sustain contraction, and you're going to need ATP to relax. These are very energy needy. That's why I say to people, if you're looking to lose weight in any program, you better exercise, because the more muscle you build, the more energy you will need even at rest. Because these are very demanding tissues metabolically. Is your question, Kim?
Yeah, I was like, that was just a skeletal, or because I was like, I thought that like cardiac, even if you had ATP, if you didn't have potassium, or if you didn't have SA or AV nodes.
Well, it's gonna see now in cardiac, we're gonna have mechanisms to avoid
so
that.
Okay, so it's
right.
not about the.
So if you didn't realize this, cardiac muscle, of course, we got to back this up to something we didn't cover in a great extent here, but we're going to cover again. In this chapter, and you're going to see it again and again, the idea of the mitochondria, right? So that's all kinds of mitochondria here. See that, right? We have many nuclei, but we have lots of mitochondria. And this is true. They showed us this little purple squiggle here, and they show it, back in your one cell here. So you guys know on some level that mitochondria, as we talked about organelles, do, right, our energy, our cell respiration. This is the site, right, where we get the most efficient transfer of energy from macromolecules like amino acids, glucose, fatty acids, to ATP. And we have this muscle, this cell that really demands a lot of ATP. So, by the way, it has a lot of mitochondria. All right? Now, heart muscle has 10 times more mitochondria per cell than a skeletal muscle. So we got to have mechanisms to prevent this possibility of fatigue.
So if you
And
take
you-
in too
you've
much
evolved.
calcium and too much, then that other ones might not.
Yeah, if you, but again, that mitochondria is just there for the ATP, right?
Yeah.
And heart muscle is very good at using, and skeletal muscle is too, using other sources of energy, not just glucose.
Mhm.
They're very, very good and actually even prefer fatty acids at rest.
So if your heart muscle fatigues, then you're gonna die.
Yeah, because you won't get contraction, right?
That's the reason
And that-
people die?
Yeah. So generally what they've done is they've lost muscle cells because we've already said that these guys don't actually undergo mitosis, okay? So if you kill heart cells, and there's many ways to do that, or muscle cells, they won't replace themselves, right? They'll try to compensate for the problem, but they won't replace themselves, all right? So ultimately when somebody dies to a heart-related issue, it's because you just can't, right, push any more blood. You're killing the whole body ultimately, and the heart, when the heart stops pushing, everybody dies in the body because this is the pump to move the blood.
So it's like the electron transport chain would be like the most destructive thing.
So when I ask you about relaxation, I'm going to give you, here's a couple of events we're going to need. We got to have ATP. We're going to get rid of the chemical messenger though, right? So that's why this was a choice. If I took acetylcholine to tell it to contract, I need to remove acetylcholine, right? If I want to stop the contraction and get relaxation, don't I? Well, even if you don't buy that, yes, that's the way it works, all right? I got to pump all this calcium that we're going to talk about and give it its role that we've already said binds to troponin. I got to pump it all back in. That's where the ATP comes from, right? The ATP helps unbind the myosin and actin, and it helps move the calcium back into all this sarcoplasmic reticulum, right? Shown in white here, shown in yellow there, but these are the tubes where I store calcium. Because I only want calcium to come out and play, right, when I'm ready to do a contraction. So, it wouldn't be leaving, because that would be a contraction. They wouldn't be opening, because that would lead to a contraction. We're going to put these in steps, and of course, no. So, I'm going to get rid of that acetylcholine, and that's going to be one of the first things that stop my contraction, start my relaxation. Again, not surprised here, which is used to store energy. A lot of people took ATP, but I want you to understand ATP is not a storage form. I don't care what you read. This is an unstable molecule that will not stay together for a very long time at all, I mean seconds. So we don't classify it as a storage molecule for energy. It is a form of energy, chemical energy, we have to get to. It is absolutely required. But you can't store that. And that's why I said, when you take in enough energy, say you had a really good breakfast today, right? And you've satisfied your energy needs, your muscle needs going to be prominent, and your nervous tissue needs. If some of you are kind of dozing off there, kind of zoned out, so I get, you probably satisfied your needs. All right, I'm not really stimulating you, right? And those that are just kind of slouched over or stuck in a place, not sitting upright, you've satisfied your needs. So you're not going to keep making a whole lot of ATP because it'd be wasted. So you're going to change your biochemical pathway and put it someplace. Now for long term, you're going to stick it here. Hands down, skeletal muscle is a place we put this molecule glycogen. And so is the liver. But again, I said can be used, released from the mitochondria in the muscle, it's going to be this. Creatine phosphate is going to be our choice. All right? Glycogen will be stored long term, all right? But here's the first place I'm going to stick it. And that's kind of what I said. You kind of have to rank things a little bit, all right? And try to understand where the question's coming from. Yeah, Tanner?
What happens when you drink an energy drink? Like, does it just make more ATP?
No.
It's fat, yeah.
It probably goes to fat if you don't do anything, honestly. I mean, it's energy still coming from the calories, the carbohydrates, the lipids, and the proteins. And some are easier to access digestive-wise for absorption than others, and that might be what they're trying to, really sell to you. But once those molecules are absorbed and go in the body, well, they go anywhere you need them, right? And your body is biochemically programmed. And we all have different rates of this. There's variations in this, but once we have satisfied, again, our need for this molecule, and we always have this need, by the way, but it changes in amount. So once we're stable in our needs for ATP, right, we're going to look to stick that excess energy in some of the molecules for a long-term storage. And that's part of your survival, right? Because otherwise, if you didn't have this kind of strategy, you guys would be forever out there foraging for food because you would have a constant need for energy. And while it might sound really attractive to eat, and not gain weight, because that's how you gain weight, is you store the energy. It's a mass, right? Matter leads to mass, right? It would be highly detrimental for the species as a whole. So it's much better to be able to store and survive to the next time you find food than it is to spend that immediately. Did you have a question, Katie?
Oh no, I was gonna say I did research on caffeine once from energy drinks, and whatever, there's this chemical in it, or this molecule in it, that whenever it tries to enter your brain, it acts just like a different chemical. I don't remember any of the names of it, but it looks just like another chemical that tells your brain that you're awake. So it doesn't actually give you energy, except
No,
for the
no.
sugar.
Caffeine has no calories in it.
But,
Yeah, you always have to look for calories. And then, again, with the brain it gets complicated because it depends on where the target is, if you can cross the blood-brain barrier and so forth. So, and again, this might be a new term for you guys, but get used to it. So see these phosphates, these are key. They're key to being able to quickly transfer energy. So as soon as you've met your ATP needs, and again, if you had a good breakfast, that's where you're at. You are going to, in all the skeletal muscle in your body, you're going to make a little creatine phosphate. And then once you've stored as much creatine phosphate you can in this muscle cell, you're going to start making some glycogen. And then when you've stored as much glycogen as you can, it's all going to fat, because it all does. Doesn't matter what the calorie came in. If it's an energy unit, we can convert it to fat. And that, again, is really good news for the species, unless that species happens to live near grocery stores and restaurants and doesn't do a whole lot of work, right? That's when it becomes bad news for the species, right? So this is, again, try on some level, thinking about this, reflect on this, because that's how you're going to get this. Muscle fatigues, again, we'll talk about this. We do stop, again, as we're trying to keep on going, because that's what we want to remember. We're trying to keep our contraction, even though, because we don't want to go into physiological contraction. So how are we going to deal with that? Again, it's all biochemistry, but you can relate to it. All right. So fatigues, you're not producing more ATP than you can use. In fact, it's probably you're using more ATP, right, than you can produce. All right. You're going to really switch to an anaerobic, possibly in muscle. This happens, possibly. And if you do that, then you get lactic acid. So this does happen, and that's the soreness we often associate with, is somewhat due to lactic acid. All right? It's running out of myoglobin, so some of you who wrote this, again, myoglobin, we're going to decide what that molecule is, because this is a very real and a very important molecule for us. All right? And no, the neuron's got plenty of acetylcholine. Right? It recycles acetylcholine all the time. That's not going to be the likely reason why you're starting your contractions get fatigued. Contractions are weakening, and they're going to stop if you don't change the circumstance. That's fatigue. All right? So what is a threshold stimulus? Again, most people got this right, but there's a minimum requirement for how much acetylcholine gets released and how many sodium channels open. There is a physical requirement for that. All right? But it starts with that. If a nerve cell, a neuron does not release enough acetylcholine, you won't get a contraction. Okay? So we want to be very much involved in this idea about how does this process step by step go. All right? This one was really bad. I mean, recruitment doesn't surprise me. And again, increase in number of motor units. Yeah, you're recruiting friends. I always like this, right? Trying to move, right? You move from one place to another place. It takes a lot of effort. You usually go and ask some friends. Well, the same thing happens in you guys, right? Right? So that motor unit may be 2 to 1,000, right, cells, but then you have tons of motor units on top of that within these muscles. And the larger the muscle is, the more likely it's going to have a bunch of motor units. And of course, when you build it and make it larger, it develops this. Okay? So when I need more force, And I can't do that as a one motor unit getting excited. I'm just going to get other motor units excited. And we're going to call that recruitment, because that's exactly what it is. I'm recruiting some friends to help me do this. Okay, what's a good example? Have you ever been in that case, right, where you were trying to pick something up and you just couldn't move it? Well, you've tapped all your friends. All right, so what do you just can't move it, right? It's just too heavy. But you're trying, aren't you? Okay, so you recruited. Another good way to think about this is like, so if I want to move this, and I'll put it back, I promise, right? There's so much force I have to generate to move that up, right? I have to overcome gravity and the mass and the effective gravity on this, I get it up, right? I certainly am going to need a little bit more here, aren't I? Especially if Alex jumps on the table, right? I'm going to need a little bit more. But I'm using the same basic muscle groups. Well, the difference is, again, I've recruited, right? I didn't need as many friends to move this. I needed a whole lot more to do this. I call that process recruitment. And I'm just explaining that every muscle, like everything in your body, has a limit to how much it can do. All right? Tectonic, we've got to understand again, these are all going to matter. They're all terms we're going to use. Summation, concentric, tectonic, we're going to just define those. So they're not just distractors, if you will. All right? Again, not well received. This was, again, what of the following would be an example of isometric? Not surprised. Iso is equal, like it always has been. Metric is measure. So when you have an isometric contraction, you are not lengthening or shortening after a point. You're sustaining the length of the muscle. Okay? What's a good isometric contraction in you? What's one where you're not really lengthening or shortening the muscle, but you're contracted? You're not relaxed. Please understand, a lot of your muscles don't. So you're dead, right? So what's one that's good? Aren't you all sitting upright? It's your posture muscles. Your heads are upright. Isn't the tendency for them to go one way or the other, right? Well, you're holding them at a length, aren't you? Your back is straight. If you're sitting there with flexed arms, that's an isometric contraction, isn't it? Isn't it? I mean, otherwise I see you guys doing all this kind of nice stuff, right? And I don't, right? So, I mean, think about what we're talking about here and think about how that works. Okay, so we'll have to define that, which is following is true. So that the chapter ends with this idea of slow twitch fibers, fast twitch fibers, right? They call them oxidative though. So let's stick to those words. So there's slow oxidative, fast oxidative, and fast glycolytic. And what those terms are trying to tell you is about the source of energy, right? Because if it's glycolytic, it's using glucose. It's using glycogen and glucose. And that's, right? We're very limited in our supply of energy, but that's quick energy. So I hope to convince you at some point that those fast glycolytic, those are like your sprinter muscles, right? Sometimes they're like eye muscles. When I move something very, very quickly, that's probably a fast. And fast is referring to the rate of, how quickly I can get it to contract. The glycolytic is referring to its oxygen source. So oxidative tend to rely more on full cellular respiration. So a glycolytic would probably be more glycolysis, right? A quick anaerobic respiration. An oxidative, again, acknowledging the oxygen, is going to be one that I'm going to really want a lot of mitochondria, so that's a hint for the difference, all right? So fast glycolytic are not going to have nearly as many mitochondria as slow. Slow oxidative are going to have a lot, aren't they? Right? Yes, sir.
Is there a way to extend the fast glycolytic process a little longer? Because it seems to always be really short comparatively to the other ones.
Not that I'm aware of, because it's genetic, all right? And so we even debate, your book does talk about this, because your book has a lot of nice vignettes, are people just born with these differences, or do they have a lot of ability to develop them? And we don't know for sure. We know that we can influence these fibers, but could we take somebody who wasn't a really good marathoner, and make them the winner? Yeah, could we do that? And I would be leaning towards maybe not, honestly. All right, I think we all again, and it just has to do with limitations in genetics, because this is Yep.
They've got it now in 7 generations now.
So it's that's the question here. So then I ended up asking some questions about smooth versus cardiac, which you want to be aware of. Because they all three move, they have similarities, and then they have really big differences. Okay? So we've talked about the fatigue, we'll talk about mitochondria, we'll talk about the patterns of organization, and a few other things about those muscles. Okay? Depolarization. This term is never going to go away. All right? So depolarization of the sarcolemma. So back out again. I got to remember that sarcolemma is a membrane. So what is it's a change in the electrical properties across the membrane. So from outside and inside. It's right at the membrane of muscle cells and nervous tissue cells, neurons, right? Because we call both of those types of cells excitable. We did not call epithelial cells excitable. I wouldn't call most of you guys in here very excitable right now, but right, connective tissue, not excitable. Right? This stuff, excitable. And that's because I can influence the electrical charge. So when I say something is polar, what am I telling you? When I say this is a polar molecule, like water, what have I told you?
It has a charge.
Yeah, it has both, right? A positive and negative, doesn't it? You've got this charge, both charge presence, and they're separated on this molecule, right? So I'm still talking about the same basic idea, but not on a molecule alone, right? So here, when I say it's depolarizing, well, the first thing I need to understand is that in a cell at rest, right? And I said it's excitable, so it means I can get it out of rest. When I say excitable, right? As I was talking about you guys not all looking very excitable because you're kind of in a rest state, right? I'm not trying to really honestly, I don't want to monopolize the conversation. You all feel to jump in. I'm just trying to say, how can you relate to this without just looking at these PowerPoints? Right? So, right, at rest, muscles not doing anything. What is the situation? Well, the situation is that, right, here's our membrane, and let's just do it as a cell like this. Let's just make it, here's our membrane, okay? So there's stuff in here, the cytoplasm, and there's fluids out here, right, which we call the interstitial fluids. And they're always there, right? Cells are bathed in fluids, and then their fluids are compartmentalized in blood vessels and lymph vessels and cerebrospinal fluid. I mean, you're just a bunch of fluids, all right, with these cells stuck in them, all right? So there's these charged, right? We say polar is a difference in charge. So there's these charged ions, right? That's the term we use for them, that are out here and in here, okay? And they're not the same ones. And we know why, don't we? Because that membrane, right, with its phospholipids, doesn't allow anything with a charge, anything that's polar, to move across the membrane, does it? It lets fats go because of the nature of the phospholipids. We said this membrane is a great barrier. This membrane contains a cell, it defines a cell, it determines what will happen to the cell. It's a gatekeeper. And we said it's a gatekeeper because it has all these different channels and receptors and everything in it. But without those, we're not moving anything that's charged. And you notice I said channel, right? And we're going to talk about these sodium channels in here a lot. Well, they're going to be scattered throughout here. Okay, I'll be right with you, Kale, right? So I can move ions, but I have control, don't I? I'm not moving them without something happening because the membrane is phospholipids. All right, everybody there? So this means there are certain ones out here that can't just automatically come in here because if they could, right, we know from diffusion they would. And the inside would be the same as the outside. Because that's what diffusion dictates, doesn't it? That molecules will move until their concentrations are equal. But then I can't do what I need to do here. I promise you, it won't work. And if they come to equilibrium, you're going to die. So I need this difference. And here's all you need to understand, because there's a lot of stuff out here, a lot of stuff in this solution. There's a lot of stuff in this solution. Here's what you need to hold on to. That sodium ions are very high extracellular, and never forget that. That is really important to life. Very, very high outside the cell. And they're not getting in, all right? Potassium is very, very high inside the cell. And it's not getting out on its own. There's a little bit of leakage that happens, but this is important. And this is not going to change in anything we talk about. So you want to take baby steps here, right? Again, this is extracellular outside the cell. This is inside the cell, potassium. Sodium outside. Okay? So now there's a more complex process than your book, or certainly more than the whole book talks about. But here's what happens in a cell, again, at rest, not contracting muscle, not excited, not being told to do anything by a nerve. Well, it turns out that a little bit of potassium tends to leak out, because you have a lot of different types of channels here, okay? And so over time, spontaneously, there's a little bit of positive charge that builds up all around this cell, right at the membrane. And because this positive is leaving, it is negative inside. There's our polarity, isn't it? That's a resting membrane potential, and that's a very important word. And again, if you don't get all the molecular mechanisms, that's not a big deal. But you need to grasp this concept, or this is going to be memorizing different things, and eventually you're not going to keep them straight. You can't, I promise you. You got to relate to them. So when acetylcholine comes along, right, and binds, right, so here's a little, let's draw this receptor, and here's our neuron up here, coming down. I know it's not a great model, all right, so, and it releases acetylcholine, which we always abbreviate like this, and it binds here, that's our little acetylcholine vesicle. Well, this is actually associated with one of these channels, which is a sodium channel that we generally call a ligand-gated channel. All right, this relationship is extremely important. Once this event happens, it will open this channel that was closed, right? And being closed, that stayed out from in here. But when this opens, what's going to happen? Sodium's going to flood in as much as it can in this short period of time, right? Because it's high outside compared to inside. What's going to happen to the charge? Because this is the important part. What happens to the charge? Yeah, it'll depolarize, right? Yeah, you go away from polarity. It'll start increasing, won't it? Start becoming more positive. And more positive and more positive. Okay? So depolarization means, right, the inside of the membrane has become less negative as sodium moves in. Period. That's what we want you to grasp at this level. This isn't really hard to relate to. Just again, it's just something you don't see. But it's not a hard concept, I promise you. All right? Okay, which of the following statements is false? And this has, again, to do with the slow oxidative fibers versus, and I wrote slow twitch, so my apologies. Twitch is a little bit less specific, but, all right? And so that's just, we're going to define that. And according to the sliding filament model, right, so again, remembering that the filaments are actin and myosin, right? Binding sites on actin open when, right, calcium. So we're going to come back and we're going to look to give a role to all these things. All right? So, you sufficiently wondering? So, again, having said that, I've given you a lot of background here. I've spent, when we start the class at 9:30, I've spent an hour just talking about questions and trying to get you to understand why those questions were there on your quiz and why they were difficult. Because there wasn't a buy-in yet because it doesn't count, but there's also, it's conceptual stuff that it's a visualization. So I'm not going to hang around. So if you're one of the students that writes down everything I have on a PowerPoint, please see me after class. And I got to go to a lab after class today, but see me today sometime after 1 o'clock, and I'll print you the PowerPoints. That's not the important part. The important part is, again, that hopefully you know, as you guys have been on your phones, maybe been recording me talking this whole time, right? And you're going to get all this because you got to spend some time. And it's not just going to be with me, right? You know, you got a limited time with me. It's going to be outside this class. But if you invest this time and try to grasp what is happening in you as well as everybody else in this room, you're going to be much better prepared as we move on and talk a lot more through physiology classes about how does the body work. All right? So again, I promised you we always got to tie you back to homeostasis. So keeping everything moving. That was our example of the heart tissue, right? Your heart stops contracting, blood is not moving. Every cell in your body is starving to death. Rapidly or slowly. You take your pick, but it's going to starve to death, all right? Because it needs that oxygen from the blood, and it needs those nutrients from the blood. All right? And then, of course, you keep your food substance, your chyme moving in the small intestine. You keep gases moving in the respiratory system. You name it. And then, of course, you're going to be studying these skeletal muscles, thinking about them moving, right? Maintaining posture, blood, lymph, chyme, gases, etc. But I want that grasp. All right? So you did this, and again, in capitals, so you get this excitability. They all have to be able to respond to nervous. Now, right away in this chapter, they point something out that is extremely important and throws so many of my students, and I don't know why, I can't even guess anymore, you know, because I get too old, right? Is that skeletal muscle is only going to be responding to a motor neuron. Pretty much, that's their relationship. And that's where we get the motor units. Cardiac muscle and smooth muscle actually contract for other reasons besides the nervous system. Right? So cardiac and smooth muscle can respond to hormones, and they can respond to local changes in ions and molecules. So they have a lot, maybe finer and more varied control. That's why it's nice for us to start naming people on the skeletal muscle, because it's the easiest one to understand. Okay? So that contractility is a unique characteristic. I didn't mention that in epithelial. I didn't mention that in connective because they didn't do that. They didn't need to. Right? This is a muscle thing. Right? And these are the key players, but not the only players. All right? Elastic and extensible. So this means, yeah, I can stretch it and it'll go back, right? And it does, by the way. Muscle's got a certain stretch to it. And, right, these two, they're different things, but they really are important characteristics that work together. So this is what they've got in common. So where are the different? Speed of contraction, right? So skeletal muscle is the fastest. Hands down. Organization of these proteins, like actin and myosin. So you guys realize from studying the tissues, right, that cardiac muscle and skeletal muscle, right, have this very similar striations. So they actually, sarcomeres are common to both, and they're organized the same way. You couldn't tell the difference if you looked at an electron micrograph. All right? Number of nuclei. Well, cardiac and smooth, right, have one and only one. Skeletal, we said, has many. It's those purple dots. It's always purple when it's hematoxin eosin. And how they become excited. As I said, smooth and cardiac will respond to nervous tissue. Nervous tissue can tell them what to do, that's for sure. But they don't require, right? Some smooth muscle is really cool because it is so varied in what it responds to. Stretching a smooth muscle will just get it to contract, right? Just simply doing that, right? Extensing, sensibility. If you stretch a smooth muscle, it'll contract automatically. It doesn't need a nervous system or nervous cell to tell it to do that. All right, so as we look at these functions, right, yeah, you get this. We always have to start off with the whole chapter saying, okay, here's what we could do. Probably the only one that might respond, surprise, is like that one. But again, as we said, these guys are so metabolically needy, right? And every time energy is transferred, heat is produced. That's the second law of thermodynamics. So your internal body heat comes mostly from your muscle. Which is why, you know, when you're really cold, we might even get a little shivering here. I mean, we're trying to generate heat in that circumstance. So they maintain this because every time they're transferring that potential energy from glucose or fatty acids or amino acids even, right, to ATP, right, there's going to be heat that's generated. So they would be that responsibility. So this should all be, I hope, pretty easy to understand. Okay, so here we start to go into these specialized terms. So this stuff, again, is just kind of background. Here's what we need to know. So here's our cartoons. We use these muscles like this. But these, again, remember, these are just the cell. So we have to back up and say, okay, well, what do I see in a muscle? And so I say to students again, look, man, if you eat meat, take advantage of your own habit and examine that. Most cuts of red meat, and because of the contrast in the red, you can really see this, you can see these connective tissues that surround them. If you're eating chicken, it might not be as obvious, but you can still see it because there's a different in texture. You know, when I go in and dissect a whole organism, I'm dissecting it on the basis of the textures, not the basis of color, because there isn't any real big differences in color. It's all from hemoglobin in the body, or some derivative of hemoglobin like myoglobin. So, again, it's these textures. So you're going to see this really thin connective tissue they peel back from here, surrounding the big muscle. So whatever muscle you start to learn the name of, like Alex, you asked me about the masseters, and you guys probably learned the orbicularis, right? Oculi and oris and the zygomaticus, okay? Does that sound familiar to some of you? Maybe the temporalis, because they probably did chewing for you already. Right? Well, that's the whole muscle, and it is definitely separated from all the other muscles. You don't see that on your models here, but you know it's going to be there. And it's attached. So if it's attached through a band, that's a tendon. If it's attached through a sheet, well, that's an aponeurosis, which is also what you see here on the skull as well. Okay? So then I start to delve in. So this, again, epi on top of mysium muscle. So this epimysium, I kind of peel it back, and I get to this organized muscle. I said, so it's organized into these bundles that we call fascicles. And I said, that's what you see. You can see that with your eyes, these fascicles. If I look and zoom in on a fascicle, right, one fascicle, it's made up of all these muscle fibers, right, or cells. So one fascicle would be a whole lot of cells, in which, again, on your models, you only have one. So then they pull this out so we can look, right, at that. And so as I look at this muscle fiber, right, then first I'm going to back up and say, okay, so each of these, right, fascicles is separated from each other by a peri, because peri's around, perimysium. So epi around the whole thing, and then within it, dividing it into fascicles is a peri, right, which you see here when we look at just the fascicle. And then each of these individual muscle cells, right, they're separated by this most internal, which is the endomysium, okay? So on this model, it's still there. If you didn't realize that, if you haven't looked at this, right, this model, that's the endomysium. Right? It's going to separate each of the individual, right, muscle cells or fibers from each other within that fascicle. So it's still attached, okay? So, and then here's, right, we're getting deeper in here because we want to get down to that sarcomere, right, which is the smallest possible unit, right? So here, as I look at the cell itself, which I can also call a fiber, I got a sarcolemma, right? And that's evident on these, right? It's the same thing as the plasma membrane. So we just call it the sarcolemma because the muscle gets a lot of special attention. It's a very unique tissue, right? So now I start to look at the actual cells, right? Sarcolemma, sarcoplasm. So sarc, a lot means flesh again. So that's the cytoplasm, that's the membrane. Sarcoplasmic reticulum is just smooth endoplasmic reticulum, right? But it's going to specialize in calcium storage. So very, very important role for calcium. I know I haven't mentioned it here, but you're going to get it. Right. T-tubules that we talked about, right? These invaginations, it's part of the sarcolemma coming into the cell. So here's again, we've really telescoped, right? We've really zoomed in here in this picture. So here's still the whole cell. Myofibrils, well, that's what all these are, right? And that's why I said you've got like maybe 7 or 8 myofibrils on a model like this, right? You've got like 1.5 on that one, okay? But myofibrils, and the myofibrils are made of myofilaments, which the important ones are actinomycin. I shouldn't say important. The most prevalent ones are actinomycin. All right? And we really want to understand that sarcomere. All right? So here's where we want to be, right? So a sarcomere is a Z line to Z line. So I said take the walls of this room. We can go this way or this way, and you are a sarcomere, right? Now, we have to understand that attached to those Z lines that I show you here and here are going to be the actin myofilaments. Okay, so they're going to be attached to those. All right? And then we're going to have these myosin, which they are physically, there's more matter to them, so they're thicker, larger, denser. So myosin is easy to detect from actin, and they just kind of show it as such on a model. And it's a bigger molecule. And on this model, right, they just show it lines, right? Really, really dark lines is myosin, and the thinner pencil, really, really thin lines, well, that's actin. And we know they're key to getting this muscle to do its job. All right, so we really have to understand the organization. So dark and light because of density, right? So the A band is an area where actin and myosin overlap. And it tends to be right big area in the middle of the sarcomere. And then the I band is, it actually spans a Z line. So the I band is actin only. I'm sorry, I wish the A band was actin only, right? Because I could say to you A for A, but that's not the way it works, right? So I, the lighter band, right, is actin only in majority, and it actually spans a Z line. So a Z line is right in the middle of an I band. Yeah, Kale?
So, two things, are the lines are that, is that there for strength and stability overall? Like the more lines you have, essentially there's going to be strength and stability, and...
Well, you know, I mean, when you want strength and stability, actually just add more and more of these myofilaments.
Well, that's what I'm figuring out. So if you have multiple types, you would allow more
mechanistically,
strength, but
I can't say that it is or isn't, honestly. I just can't. But I'm sure like a biophysics might look at that and say that. But again, you got to remember that when you're delving down to that, well, you have to be able to extrapolate back out to the organism.
Yeah.
So, all right. So your H zone is actually an area where there's not actin. And it tends to be right in the middle because there's our sarcomere. H zone right in the middle. It's where there's myosin only. So the actin is stopped. All right? And then the M line is actually a structure like the Z line for anchoring the myosin and other proteins. Anchoring, keeping them in place. Because again, these aren't floating all over in the sarcoplasm. They're actually held in place. So, all right, here if we zoom in and then zoom in some more, we really get a good idea. And that's why you have that other model that you guys use in lab of just the sarcomere. And please do pay attention to that. Okay, so here they're trying to show again, here's our Z line, so here's our sarcomere, right? Here's that's our I band, A band, right? Zone, when it's just, so zone when it's just myosin, and right in the middle, okay? So let's magnify that up. And if we did, myosin has this very unique, right, shape, because we know proteins will. Proteins vary in their shapes, and that's why they can vary in their functions. So myosin has got what we call these heads. They kind of look like golf clubs, really, when you look at them, right? They got all these heads that usually stick out as like this dimer type appearance to them. And they're very tightly wound together. So the number of these heads really all give the potential for binding. And the more binding you can have between actin and myosin, the more force you can generate, right? Because the force begins when myosin and actin bind. So that is an important, physical attribute of the sarcomere. All right. When we look at actin, actin is a dimer, which means it's two polypeptides that are globular, so little balls like, and they intertwine. So people often describe actin as looking like strands of pearls. And again, they do when you look at the electron microscopy, but this is an important aspect of it. So when you look at actin, it's got that appearance, and again, it's less matter, so it's a thinner line, okay? And again, there is overlap between them, but that's if I just look at this. Now, on this actin are a few other things that we have to know about in this class. We have to know about troponin, we have to know about tropomyosin. We already defined in that quiz that troponin is going to be the calcium binder. This is important for calcium binding. The tropomyosin is going to be a kind of rope-like, similar to collagen, that covers, see how it's running right along that actin. And so if I zoom in a little bit more here, I realize that, right, tropomyosin is covering an area on the actin, a binding site, for myosin. So the tropomyosin is in the way. So in this situation, actin and myosin can't even bind. There can't be a contraction, right? Because tropomyosin's in the way. It's blocking the physical ability of these two molecules to contact. So how am I going to move tropomyosin? Because I need this to bind to that, right? These little sites here are where those heads are going to bind. And if I don't have that happen, I'm not contracting, okay? So, and again, they show this. They also show you that there's an ATP binding site. Yeah, hint, hint, because ATP is really important here, right? And ATP is going to move this head. And when this head moves, if it's attached to this, it's going to move this, isn't it? And if it moves this actin, it's going to move the sarcomere, isn't it? Yeah. So that's what I want to be thinking about. So we're going to look at that mechanism of contraction. We're out of time today, but I want to give everybody a job. So again, in your minds, today and forever, troponin, calcium binder, tropomyosin, covering binding sites for myosin on actin. Actin and myosin, they're going to be the big deal in a little bit here, okay? And we'll move forward into this, right? And please let me know again if you need a PowerPoint